So we study memory, and we were interested in how it's formed and stored. Memory is central to our sense of self. It's how we accumulate experience and become who we are. And while we may tend to view memories as intangible, they have a specific physical substrate. And that's what my students and I are interested in studying. So why do we care about memory? Well, it's an important basic science question, and it's really very interesting. But it's also fundamental to an understanding of who we are and how our brains function. We make assumptions about how our memory works all the time, but these assumptions can have rather serious consequences. And the reliance on eyewitness testimony, for example, in our judicial system is one example of this. We also want to know how to fix memory when it breaks. People are deeply affected when our memory doesn't work the way it's supposed to. While we see a gradual decrease in memory as a function of normal aging, memory impairment is often an early sign of neurodegenerative disorders like Alzheimer's disease. And as we get older as a population, this is becoming a real crisis in need of effective interventions. Losing your memory is bad, but we can also have problems if we remember too much. Current work on post-traumatic stress disorder suggests that here, strong emotional memories related to trauma can't be suppressed or are stored in some way that defies regulation. So there's as much interest in erasing memories that we don't want as there is in preserving memories that we do. So the first key point that I want you to walk away with is that each of your memories represents a physical change in your brain. Brain cells, called neurons, are plastic or changeable, and your experiences drive neural activity that induce storage of memory traces in the brain. The change can become essentially permanent, allowing you to store information for your whole life. So, about 86 billion neurons in an adult brain with an average of over 10,000 complex connections to other cells. How do we go about finding the needle in the haystack? The answer is to leverage the strength of experimental psychology with tools from molecular biology, genetics, and imaging. We can narrow the search because we know a little bit about patients and people with brain lesions. We know some areas are particularly important, but there is no single memory center in the brain. Memory takes many forms, and the circuits that you use to remember how to ride a bicycle are very different from the circuits that you're going to use to find your car when you leave tonight. We can also monitor brain function while people perform a memory task. In our experiments, we measure a signal that tells us how hard the cells in the brain are working and use this as our way to figure out which parts of the brain are important. This is a very popular type of study, but there are real limitations with this for getting down to the nuts and bolts of what's actually happening in the brain. If we focus on one brain area, we can use genetic strategies to label or tag groups of cells that encode a particular memory, since active cells express different genes than cells that are not active. So we create two memories, maybe two different mazes for the animal, and then take the brain out and look at where the signal for one set of cells active at time A and one set of cells at time B might be. Well, that's fine, but that's kind of a correlational procedure. What can we do to actually infer cause and effect relationships? What if we could control the activity of the cells that encode a memory? Well, it turns out that we can do that. And there's a couple of techniques that we can use to do that. One really popular one now is called optogenetics, where we put in a transgene that makes the cells express a light-sensitive ion channel. And once they express this channel, we can shine laser light of different wavelengths onto the cells and turn them on or off at will. And several recent experiments have supported the idea that activating a particular population of neurons in this way will actually cause an animal to retrieve an established memory and behave accordingly. But the real action in memory is at the synapse, or the connection, the communication between cells. We have 86 billion neurons. We have 86 trillion synapses, which makes the brain, sorry, Paul, the most powerful supercomputer in the world. So we've known for a long time that learning changes synaptic physiology, and it changes synaptic structure. So this idea of synaptic plasticity is central to our understanding of learning. There's various ways that we can measure this. But the key to synaptic plasticity is the altered gene expression and subsequent changes in local protein synthesis that happens in neurons. These new proteins allow for alterations in both cellular structure and cellular function. Now, we've looked at protein synthesis in my lab since the 1990s, but we've recently started to focus more on a related process called protein degradation. You probably didn't know that your cells have a biological garbage disposal called the Ubiquitin-Proteasome System, whose job it is, or UPS for short, whose job it is to break down and get rid of proteins that are toxic or no longer needed. The system does a lot of basic housekeeping and protein homeostasis, but we've recently shown that the activity generated when you learn turns on this system, and if we prevent the system from turning on, we prevent the storage of memory. So the idea is that before you can strengthen a synapse, you have to break it down and destabilize it. And this destabilization is what the UPS does. This is critical for our understanding not only of memory formation, but of forgetting and memory modification. The UPS is also interesting for other reasons, because one thing that we've noticed is that protein degradation, or a decrease in protein degradation, is part of the normal cellular aging process. And Alzheimer's disease partially involves an inability of cells to clear toxic proteins. So we did a few experiments to look at this. We looked at young rats versus old rats, and we found that young rats have good memory and normal UPS function, whereas old rats have poor memory and depressed UPS function. And importantly, the rate of protein clearance actually predicted the memory impairments in the old rats. So that's kind of cool. But really what we want to be able to do is target this system for interventions. We've tried a couple of ways to upregulate UPS activity to fix the memory problem. We haven't had any success yet. But right now, we're trying this promising new technique of gene editing to actually reprogram the UPS, make old neurons look like new neurons, and maybe fix the memory problem. So the last thing I want to say is that none of this work would be possible without UWM PhD students and postdoctoral fellows. I've had some great students over the years, and they keep me excited, and they do all the hard work. And my final thought is support federal research funding, because federal research funding enables us to do this kind of work, and it also is going to allow us to have a prosperous and happy future. Thank you.